Hi, this is Dr. Collins Jones. Let's talk a little bit about genome editing in the clinic. And in this particular application, we're going to look at a way to help manage HIV in patients. In this particular case, what we're going to do to edit the genome is we're going to remove T cells from the patient. And those T cells, of course, are the reservoir for HIV, for AIDS virus. It turns out in order for HIV to infect cells, it actually uses a receptor called CCR5 as well as the T cell receptor CD4. And so what we're going to do is if we remove one of those receptors, then the virus will no longer be able to infect the cells. And we're going to be able to achieve that by using something called zinc finger editing, ZFN. And so with zinc finger editing, we can target what sequences of DNA we specifically want to remove. This is different than your traditional restriction enzymes. Traditional restriction enzymes are found in nature, and we had no control over what sequences we wanted them to remove. They're already pre-programmed from the bacteria that we got the restriction enzymes from. With zinc finger nucleases, we can actually design into the protein, into the nuclease enzyme, what sequence of DNA that we want to remove. So in this case, we're going to remove the sequence of DNA that codes for the protein CCR5. And so once we've done that, we then take these T cells have now been, their genes have been edited to remove the CCR5 gene, so it can no longer produce the CCR5 protein, and we now reinfuse those cells back into the patient, and the patient's T cells now have CCR5 negative, or they're CCR5 deficient in the T cells, and those T cells multiply and grow in the patient, and they can't be infected with HIV virus. If you want to learn more about genome editing, more about gene therapy and how these are being applied in the medicine of today, sign up for one of our Biotech primer courses. Thanks.